Hi, I'm Dr. Andrew Catchpole. I'm the Chief Scientific Officer at hVIVO, and I'm absolutely delighted to be joined today by Professor Guy Boivin. Guy, I wonder if you can give us a little bit more of an introduction to yourself, please.
Certainly, I'm infectious disease specialist and professor of pediatrics at Laval University, which is based in Quebec City, Canada. And my main research interests include the pathogenesis, the prevention, and the treatment of respiratory viral infection.
I know you've got a really distinguished publication records. That's really impressive. I wonder if you can give us some of what you think are your favourite highlights over those many publications that you have and your current interests.
Well, we're particularly interested in influenza, RSV, and hMPV. Consider hMPV as our little baby. So we did a lot of studies on hMPV. Obviously, after his discovery, diagnostic tests, pathogenesis, identification of small animal models, and candidate vaccines. We have completed the clinical trials. We have a trivalent vaccine focused with immunization against RSV, hMPV, and parainfluenza type 3. So hMPV and then influenza, we are particularly known in the influenza field for the understanding the mechanism of resistance of influenza to the neuraminidase inhibitors, including oseltamivir or Tamiflu. So in particular, we want to look at the fitness or viral replication or transmission of resistant viruses compared to wild-type susceptible viruses in the population. And then in the more recent years, we got interested with what we call the viral interaction or viral interference between two respiratory viruses. And we found that if you are infected by one virus, for example, influenza, and you have subsequent infection by RSV, you will decrease the viral load of this RSV infected animals compared to animals that receive only RSV. So influenza interferes with subsequent RSV replication. So we're looking at these interactions at the moment.
Excellent. I think it makes a lot of sense today, really, as we're basing this on hMPV. If you can tell us a little bit about hMPV itself and what it really is.
So hMPV is a pneumovirus, along with another well-known virus called RSV, respiratory syncytial virus. And both viruses are important causes of infection of the upper and the lower respiratory tracts of humans.
And what about the sort of disease characteristics of that? Can you describe that to us?
Well, these viruses, hMPV and RSV, are impossible to distinguish on an individual basis. You need a laboratory test to manage the two diagnoses. But both are associated with bronchiolitis and pneumonia in young children, as well as bronchitis and pneumonia in the elderly as well. And there's also these immunocompromised patients who are at high risk of developing complicated pneumonia with hMPV or RSV.
I mean, given hMPV and RSV are so similar, I'm really curious to hear about your story about how you actually co-discovered hMPV. Can you enlighten us on that, please?
Well, it's an interesting period of time. I remember hMPV was officially discovered in 2001, but at the late 90s, I had one very excellent, in fact, laboratory worker. She kept knocking at my door, telling me, "Dr. Boivin, I have something I would like to show you." And she was showing me cytopathic effects in cell culture. Remember, at that time, 25 years ago, cell culture was an important method of diagnosis. Nowadays, nobody are doing cell culture. So it's been replaced by molecular assays, but at this time, it was important. So she was showing me cytopathic effects on cells, showing that, proving that there was a virus, but we couldn't identify with immunofluorescence studies or molecular biology. We were doing in-house PCR assays at that time. So that was not influenza, that was not RSV, and that was not parainfluenza viruses. So we contacted some of my colleagues at the CDC in Atlanta decided to look more carefully on these unidentified viruses. And turns out that we did electron microscopy. We knew it was either a pneumovirus or paramyxovirus, but a new one. And so we were starting to amplify and sequence the virus in question. And just like that, there was a publication by a Dutch group led by Dr. Osterhaus on the discovery of a new virus. They call it human metapneumovirus. So then we designed the appropriate PCR primers for PCR, and bang, that was a human metapneumovirus. Almost all of these unidentified viruses were human metapneumovirus. So we were the first group to report the presence of this virus in all age groups, not only in babies, but in elderly as well.
Oh, wow. So it really was a process of elimination then, because you saw this tissue culture effect and had to then eliminate all the known viruses before you could then identify the fact you must have something new here. So did that come from, was it a clinical sample, or what was the sample?
We had many clinical samples. Most of them were respiratory tract samples from the upper respiratory tract, but some were from bronchoscopy samples and from the lower respiratory tracts. Yeah, they were all clinical samples.
Thanks, Guy. That's really good to remind us of the fact that actually we're so reliant on molecular techniques, but even so recent as 25 years ago, they weren't as routine as they are now. So very reliant on having tissue culture cytopathic effect to actually be able to identify a virus in the first place. But of course, CPE in itself actually is reliant on whether that virus is going to grow in the particular cell line. I mean, was hMPV difficult to get to grow in the cell line? Would it grow in many cell lines, or was just one or two cell lines that were growing?
That's the trick. hMPV will grow only in a few cell lines, and it takes a long period of time before you see the appearance of these cytopathic effects. Meantime, before seeing the cytopathic effects is about 17 days. And most laboratories at that time were keeping their cells for 10 to 14 days and then report negative results. So that's probably the reason why it took so long to identify hMPV, although it was not a new virus, because we set up serological assays and went back to see if people in the 1950s, we had serum collected from an old influenza study. And we went back to these samples 20 years after that, and we were able to see that all these patients in 1950 already had antibodies against human metapneumovirus. So again, it's newly discovered, but it's not a new emerging virus.
Oh, wow, right as far as back as the 1950s. Yeah, so it clearly has been circulating in human population for quite a long time. I mean, that's really fascinating, because it really does go to show how the true discoveries are always made by those scientists actually continuing to research something and actually doing something a little bit more unusual, and really the key on the observations, because like you say, a lot of tissue culture assays which are set up would be binned after a relatively short period, assuming there's nothing there. But the perseverance to leave that little bit longer really actually discovered a virus which otherwise wouldn't have been seen. So that's really quite fascinating to be able to understand that and see that's really how it was discovered. And it really does make sense of why, even though it's been in the human population for many decades, it was only really discovered in 2001.
Yeah.
So I've also, of course, it was discovered in 2001, but actually most of the general population, us non-virologists, hadn't really heard of hMPV until this year. Of course, it was in the news, in the mainstream media in earlier this year, of course, where there was an outbreak in China. I wonder if you can tell us a little bit about your interpretations of that and what you think happened.
Well, hMPV, like other respiratory viruses, will cause yearly epidemics. So during the winter-spring season, late winter-spring season, and such as influenza virus, some years are big epidemic years, some years is lesser epidemic. And what happened is that this year we had news from China and also from India and from other countries that they were fearing a new pandemic. And when the testing was done, nowadays we have multiplex PCR assays, we can test for up to 20, 30 respiratory pathogens at the same time. And it turned out that the cause for this a big epidemic that was called a pandemic was human metapneumovirus. So human metapneumovirus, again, was discovered in 2001, so almost 25 years ago. But the thing is that still nowadays, people don't know a lot of things about it. I'm talking about the general population, but also physicians. I receive calls weeks after weeks. What is that? My patient has a human metapneumovirus. Is it dangerous? So what happened again in China, there was a big epidemic of human metapneumovirus. Don't forget that these viruses will evolve over time so that we can be reinfected many times over our lifetime. So I think that we need to draw some lessons from this China experience, if I may say. We need to increase awareness about hMPV, and hopefully if we increase awareness, we'll also increase funding, research funding for this important pathogen.
Yeah, that's a really good point, because it is always really surprising to me that, like you say, even sometimes physicians haven't really heard of hMPV or don't really understand what type of disease it's going to cause. So it's even, it's been around 2 decades. So, but of course, being in the mainstream media has really meant that, just as you said, it's become almost a household name. Not quite, but certainly the general population have now heard of it. They might not fully understand it, but they've heard of it. And I think that can only be good for research, do you think?
I think so. And we still need research, especially developing vaccines and therapeutics against this newly discovered, let's say, put it that way, virus.
And I certainly have seen at hVIVO, we've seen a number of inquiries about hMPV, and prior to January, that wasn't something which was occurring very frequently at all. So I do put that down directly to the fact that there's been a lot of mainstream interest in the media, and that then trickles down into funding bodies, and just awareness and talking about a virus often does actually fortuitously come out. So in fact, you get more funding through, which is great. So obviously having an epidemic in winter is not a good thing, but actually it is nice that actually some more discoveries may come out of that, because it's put it more in the mainstream media, and the fact that that may well help some funding. So that's really good. Although it was a little concerning in the mainstream media about causing quite a bit of pandemic sort of panic, really, for want of a better word. And I wonder if you can explain to us why it is you think that happened. And of course, us virologists always understood that hMPV isn't going to cause a pandemic. But I wonder if you can explain why it is that hMPV isn't thought in virological terms to be a pandemic virus.
Well, there's a few reasons for that. One of them is that we don't know about any reservoir for human metapneumovirus, any animal reservoir for human metapneumovirus. We have avian pneumovirus, but that virus does not cross the species, so there's no human infection caused by avian pneumovirus. So again, there's a lack of animal reservoir for this pathogen. That's one reason. Another reason is that hMPV, unlike influenza, does not have a segmental RNA genome, and so that we cannot see exchange of genes between two viruses infecting the same cell, like we can see with influenza viruses. So no reassortment possibility and no recombination events, as was reported for coronaviruses. So let's say that if you're asking me what would be the next pandemic virus, I would say I would bet again influenza viruses or coronaviruses more than human metapneumovirus or RSV.
And that's because they've got such a well-established animal reservoir, I presume, is it?
Yeah, the reservoir for influenza is wild birds, as you know, and for coronavirus initially is bats, and then you can have intermediate host jumping to human.
Yeah, and it's always quite astonishing the huge quantities of viruses these animal reservoirs when actually you get an infected animal, they could be perfectly healthy, but they're still excreting huge quantities of virus. So it's, in some ways, perhaps sometimes you look at those quantities and you think it's perhaps even surprising that more of them don't jump the species barrier and get into people, but somewhat reassuring nonetheless.
Yeah, I agree with you.
So you've very eloquently explained to us why hMPV isn't really, from a virological perspective, thought to be a pandemic virus. But does that mean it's not an important virus? What's your view on that?
It doesn't mean that it's not an important virus. In fact, it's a very important virus. We know that in the pediatric population, for instance, hMPV is the second cause of bronchiolitis after RSV. In the elderly and patients who are also with underlying pulmonary or cardiac disease, hMPV is a major player. In fact, I saw some studies coming out from the United States. They have a network of university affiliated hospitals looking at the cause of hospitalization for acute respiratory tract infections over the years. And just saw a publication from that group showing that hMPV was number 3 in terms of cause of hospitalization for acute respiratory tract infection in the United States year over year. In fact, it was even more frequent than RSV and influenza in that study. So it's a major player also in that age group. So I wouldn't say that it's not an important virus because it doesn't have a pandemic potential. It's a very important virus.
Wow, second cause of bronchiolitis and a third cause of hospitalizations. Now, it really does show just how important pathogen it is and what high levels of disease burden are actually caused by this virus. So if someone contracts hMPV and say they're hospitalized, what treatments are available and how would you actually go about treating that patient?
Unfortunately, there's no approved treatment for hMPV at the moment. If I have a patient with severe disease, especially an immunocompromised patient, or a very young baby, premature baby, I would try to use ribavirin, which is a large spectrum antiviral agent that we give also in case of very severe RSV disease in immunocompromised patient. But this product has limitation. It's not a very potent antiviral against pneumoviruses. Also, is associated with a lot of side effects and is teratogenic. So it's problematic for the nurses taking care of the patient receiving this medication. So really, clearly we need an efficient antiviral agent, a small molecule that will be able to stop the replication of the virus. And this is especially important in immunocompromised patients because these patients tend to less respond to immunization with a vaccine.
Yeah, that makes sense. So there really are very limited options right now for direct treatment of these patients, unfortunately. So as you mentioned, some of the vulnerable populations can be immunocompromised individuals. So are there complications with those individuals for other infections, co-infections at the same time as hMPV, or is that not very common?
You can see co-infection with bacterial pathogen such as Streptococcus pneumoniae. You can see also co-infection with another respiratory virus. And this is an interesting field of investigation, the interaction between respiratory viruses. We realize now there's some interference between one virus and a second infection by another virus. And we're just starting to study this question of interaction and interference between respiratory viruses.
Yeah, because it's always quite fascinating to see when we screen some samples and we see that we've got these co-infections. Of course, we're just seeing snapshots in time, so it's hard to understand, which pathogen did they get first, and was they more susceptible to that second one, or as you mentioned, actually sometimes it actually prevents a secondary infection. So I think that really just shows us there's a lot more to understand in the virology field, and particularly with co-infections. Then you've got bacterial infections with viruses going on at the same time. So it really is quite an untapped area, isn't it? I think there's a lot more research needs to be done on that to help these patients.
Definitely.
You also alluded to the fact that about vaccines. I wonder if you can tell us a little bit more about where we are with vaccines, and are there any promising candidates coming through?
Well, we don't have at the moment an approved hMPV vaccine. Same thing as for the treatment. We don't have it right now, unfortunately. But there are some development. There are companies that have developed mRNA vaccines, VLP vaccines, live attenuated vaccines against hMPV, but they are in phase 1 or phase 2 or preclinical. To my knowledge, there's no product in no vaccine in phase 3 clinical trial at the moment. I think that this research on hMPV really follows the research we have seen lately on RSV vaccine as well. Since the past 2 years, there have been 3 approved RSV vaccines. So 2 subunit vaccine and 1 mRNA vaccine have been approved for RSV infection in the elderly. I must stress that it's for the elderly at the present time. So that I think that the hMPV development will benefit from the RSV story, and we're looking forward to have a vaccine against hMPV in the next few years.
Yeah, that makes sense, because of course hMPV and RSV are, as you mentioned earlier, part of the same virus family. So it's great that there's been some success in some vaccines coming through from RSV. So I'm really encouraged to hear that you also believe, like I do, that learnings from RSV and all that research will actually now help fast track hMPV development. And I know I've been in contact with some researchers who literally their whole research team has pivoted from RSV onto hMPV. So I think that's some exciting times on that, and quite very, very encouraging. At hVIVO, we are really proud actually for our small part that we played in the RSV vaccine product developments. Of course, we run human viral challenge studies, and we had human challenge studies run against RSV, and we tested a number of vaccine candidates, and that actually was the first evidence of vaccines showing efficacy in human infections, and that helped to fast track the development and really give confidence that actually this difficult to vaccinate previously pathogen, it can be possible, and these new pre-F vaccines were successful, and that then fast-tracked the development funding to push them into phase 3 and ultimately into market authorizations and where we are today. So, I mean, we were delighted that our contribution to the challenge studies could do that. Do you think a hMPV challenge model could have a similar sort of benefits?
Oh yeah, definitely. But I think also that we need to be careful in the development of vaccines against hMPV. Same thing that happened with RSV. We have not to forget the fact that we had real problems with the initial vaccination of young babies with an inactivated RSV vaccine in the 1960s. In 1969, in fact, we observed that the young infants who are immunized with the inactivated RSV vaccine, when they encounter the RSV, they develop a more severe disease than the non-immunized children. And only with the discovery by a group at NIH in the 2015, 2016, Macmillan and others, that showed that if you can fix the F protein, which is the protein that is more immunogenic for both RSV and hMPV, if you can try to fix this F protein in the pre-fusion conformation, you will expose important and immunogenic epitopes, and then you can develop safe and effective vaccines using this pre-F model, at least in the elderly. But there's still potential problem vaccinating the very young children, and we need to be careful about this enhanced disease in that small population.
Mm-hmm. Yes, because at the moment, they're effectively being vaccinated via the mother. Is that correct? So we've got maternal vaccinations.
Yeah. There's possibility of vaccinating the mother, or possibility of giving a monoclonal antibody to the young infants as well. There's two options, and then some countries have favor one option over the other.
the different age groups, of course, have got very different immune systems. Young children have got very naive immune systems, so they're not going to respond in the same way as us adults, which have been exposed to these pathogens multiple times. So it's, that makes it even more complex, doesn't it? It's not one's vaccine fits all, because their immune systems are so different, therefore they're going to respond so differently. And of course, we can look at preclinical models, but animal models, for example, the animals haven't been exposed to these pathogens, so their immune system doesn't really reflect what's going on in adults or babies in humans as well. So it's a very complex picture, isn't it? But it's still really encouraging, and that we can hear from you today about there are some candidates coming through for testing. So that's really good, encouraging news. And I really want to thank you for your time today. You've given us some really great insights, Guy. Thank you.
It was my pleasure.